So, hi, my name's Jack Greger. I go to the University of Arizona and I study bioinformatics, ecology, and evolutionary biology, which is my de facto way of making a genetics major. So, this summer, I was able to study mobile elements in plants. And we did this through studying mobile mRNA transcripts and mobile peptides. So, my project really splits into two different stories here. So for mobile RNA transcripts and grafts, when two species that are compatible are grafted together, we notice mRNA from the opposite species moving from shoot to root or root to shoot. And so we see that here, like an mRNA transcript moving to the shoot, and it's expressed there. So we can actually detect this by taking this genetic material and aligning it against the opposite genome. And so then if something from tomato pops up in this analysis, we know that it moved. However, because these species are so compatible, we notice, or because they're so compatible, they're also very genetically similar. And so this is our synteny plot, which is eggplant chromosomes and tomato chromosomes. And as we can see here, a lot of the order is retained between them. And so what this causes is a lot of genes that are ordered similarly, and also genes that are very orthologous or matching in a lot of the regions of the gene. So this creates a problem for us because we have regions of exact matching. So potentially we could have mRNA transcripts from eggplant that are showing up in our tomato analysis. So we could get false positives here. So say we did this graft and we have an eggplant root and a tomato shoot, and we detect tomato mRNA from this analysis against the tomato genome. There's a chance that those transcripts could actually be from eggplant, which is problematic for us. So we want to use areas that aren't exactly matching to perform this analysis. So what I was able to do is start characterizing these genes that do have exact matching regions so we can start moving forward and figuring out what to do with them. So part of this is quantifying what % coverage across the gene is exact matching. And so we found in 4,000 genes in tomato and around 3,000 in eggplant, that between 0 and 25% is exactly matching. And so in the future, we can use this data to start making algorithms that can filter out, let's say, below 25% or below 10%. And then we can start actually salvaging these genes that we'd normally have to discard for mobile element analysis. So in the future, we could use something like this and validate these mobile genes and then figure out what makes them mobile if we have enough data. And then we can recode these and use the mobile motifs to mobilize certain technologies. So for example, the CRISPR-Cas9 system that uses targeted gene knockouts, we could have CRISPR-Cas9 that's mobilized via an mRNA introduced into the root of a grafted plant, and it transfers itself to the shoot of the grafted plant and then makes its selected gene cuts. This is a great technology because it bypasses what something considers as a GMO. So if CRISPR-Cas9 is introduced here and is expressed here, only the eggplant root would be considered a GMO because all the transgenic material is being introduced in the root. And so this allows us to bypass a lot of restrictions that would make GMOs and studying them unavailable. One example of this could be in Florida, for example. There is a big problem in the citrus industry because of a lot of it's organic with greening. And so with greening, for example, we can knock it out, but because the industry is mostly organic, we're going to make those into GMO if we use traditional genetic methods. So if we had a technology like this, we could make those cuts, solve those problems without hurting the industry or making it GMO. So for my second project, in a similar vein, we're studying peptides this time, or small proteins that move across the plant. So the CEP system, or C-terminally encoded peptides, are a nitrogen observation response system, or a nitrogen starvation response system. So during nitrogen starvation, CEPs are expressed and they move from root to shoot, and they're received in the shoot of the plant, and another signal is sent down to forage nitrogen everywhere except where that starvation occurs. And so what we've discovered recently is CEPs doesn't just respond to nitrogen stress, but the system is cool enough and interesting enough to where we want to deduce the pathways. So what I did is I studied drought stress and how drought stress affects the CEPs pathway. So we prepared a 2 weeks old seedlings of wild type of Arabidopsis thaliana. And we subjected them to mannitol treatments at time courses of 0 and 24 and 48 hours. We collected RNA from them, reverse transcribed it into DNA so it's more stable, and then quantified it. And so what we figured out was the treatment's working because these are marker genes that respond to mannitol. So we know that the plant's responding to mannitol. And then also we found 3 SEPs that respond strongly and 2 that don't. And so what this does is starts building a map of the SEPs pathway, what certain SEPs respond to and what certain SEPs don't respond to. And so in the future, this is really just foundational work. But if we can figure out all the environmental or alternative environmental treatments that trigger SEPs, we can eliminate crosstalks between SEPs pathways. And if we isolate SEPs pathways enough, we can reprogram their inputs and outputs. An example of that in the future could be something like you have a plant that's nitrogen starved, SEPs is expressed, and we've modified the SEP receptor so that it also expresses fluorescence when it receives SEPs. So instead of waiting for a plant to visibly notice that it's nitrogen starved, we can see immediately when the SEPs is expressed fluorescence or some other detectable measure. So this technology in the future for agricultural experiments could be hugely important and make a big difference, which is what I'm excited for. And it's why I'm grateful for the Frank Lab, Lee Lab, and Boyce Thompson and Crops for all supporting me. All my friends I've made during this program that I couldn't have done it without. And so, yeah, it's extremely cool work and I'm extremely grateful.
Oh my God. Sounds like you've been really busy this summer and your projects are so fascinating. Wow, I'm going to try and stick to only a few questions, but we'll talk later. So in your first project, you talked about some potential compatibilities amongst different grafting plants. And so are only compatible plants able to move mobile RNAs across that junction point? And can you talk about that a little bit?
Yeah, so when 2 plants are grafted together, there's a graft junction. And that graft junction has to be catered to a little bit. It calluses up and then it eventually heals. And we actually have there's videos of this graft junction being healed, but if it doesn't heal properly, the vascular systems do not connect. So if the plants are not compatible, we won't get mRNA that goes across the plant.
Wow, thank you. And in response to the SEPs question or the SEPs portion of your project, so I see a couple different SEPs there. Do they all have different roles and have those quite been characterized yet?
So no, a lot of them, and that's kind of being developed right now. Now the pathways in our research, we're starting to figure out, we know how many SEPs there are, and we know a few of the things that they respond to, like a paper is recently published that relates it to immune response. So now we have immune, drought, nitrogen. We're starting to really piece it together, but a lot of that's still in the works right now.
Awesome. Thank you so much for sharing all your research. I wish you the best moving forward and heading back to your home university. Thank you so much.
Thank you.
Awesome.